Hello, my name is Dr. Stefanie Seltmann. I'm working at the Charité University Hospital in Berlin for the hPSCreg project, and today I want to talk about the representation of human pluripotent stem cell lines in the cell line ontology. First of all, I want to briefly introduce the term of human pluripotent stem cells. So after fertilization, the fertilized egg is called totipotent. That means it can give rise to any of the cell types found in an embryo, as well as extraembryonic cells like in the placenta. These totipotent stem cells then further develops into a so-called pluripotent stem cell, which can give rise to all cell types of the body, but not the placenta. That means actually it can develop into all three germ layers in the human body, the endoderm, mesoderm, and ectoderm. And in these three germ layers you find multipotent stem cells that can develop into a limited number of cell types in a particular lineage. So for example, you have the cells from the endoderm can, for example, develop into lung or pancreas, and cells from the mesoderm can develop into heart muscle cells or red blood cells. There are two kinds of pluripotent stem cells. One are the embryonic stem cells called ESCs, which are derived from the inner cell mass of a blastocyst. The blastocyst stage is usually reached 4 to 5 days post-fertilization, and at this time it consists of 50 to 150 cells. The embryonic stem cells can be found in the inner cell mass of the blastocyst. The first human ESCs have been derived in 1998. Due to their plasticity and potentially unlimited capacity of self-renewal, embryonic stem cell therapies have been proposed for regenerative medicine and tissue replacement after injury or disease. But isolating the inner cell mass from the blastocyst destroys the blastocyst, and therefore it can't develop any further into a human fetus, which raises, of course, a lot of ethical issues. Another kind of pluripotent stem cells are the induced pluripotent stem cells called hPSCs. These pluripotent stem cells can be generated directly from adult cells. The iPS technology was pioneered in the Yamanaka lab in Kyoto, and they showed in 2006 that the introduction of 4 specific genes encoding transcription factors could convert adult cells into pluripotent stem cells. In 2012, they were awarded with a Nobel Prize for this discovery. So to generate induced pluripotent stem cells, a tissue donor donates some somatic cells, and with the 4 Yamanaka factor, these somatic cells could be reprogrammed into induced pluripotent stem cells. These iPS cells could then be further genetically modified if needed, for example using the CRISPR technology, and then they can differentiate into all kinds of differentiated cell types. And these cell types then could be used, for example, for disease modeling, for drug screenings and drug development, and for toxicity tests. To get an overview on this rapidly growing research area, the human pluripotent stem cell registry hPSCreg has been created. hPSCreg provides an in-depth overview on the current status of human pluripotent stem cell research, including cell lines, publication, research projects, and clinical studies. The human pluripotent stem cell registry has been founded in 2007 to provide an overview on the research of human embryonic stem cells, and in 2009 it was expanded to also register human-induced pluripotent stem cells. Today it is one of the largest repositories for data on human pluripotent stem cells. The data in hPSCreg is entered by the user and manually validated by our validation team. The validation criteria includes ethics and pluripotency of the cell line. Today we have more than 2,600 cell lines from 31 countries. We have registered data on 136 research projects, more than 300 generators, and since March 2019 we are also starting to collect data on ongoing clinical studies using human pluripotent stem cells, and today we have almost 40 clinical studies registered at hPSCreg. But we are not only registering data on pluripotent stem cells, hPSCreg is also responsible for providing certificates for EU-funded research that use pluripotent stem cells, and all cell lines registered and validated at hPSCreg could be published in our partner stem cell research as a lab resource. As you could see here, the number of validated cell lines has steadily increased in the last year, but while the number of embryonic stem cells there are only very few increase in the number of ESCs, the number of iPSCs has grown rapidly in the last years. hPSCreg is a community project with a lot of partners around the world. After a cell line has been entered and validated, the information on the cell line is displayed public in the output of hPSCreg. So here you can find information on the donor of a cell line, general information like the name, the generator, information on the disease from the donor, information about the ASICs derivation, culture conditions, the characterization, genotyping, and genetic modifications. So far, the possibilities of ontologies in hPSCreg are only used to a very limited extent. Mainly it is used for annotation of different areas, thus diseases and phenotypes of donors are annotated, as well as disease-associated modifications of genetic modified lines. And furthermore, the original tissue or cell type and the germ layer or cell types and tissues are annotated in which the pluripotent stem cell line can differentiate. For annotation, we are using the tool Zoomer from CBI, which provides a very easy interface for a user which are usually not familiar with ontologies at all. And this tool allows us to just click on enter disease and then type in a free text, and the Zoomer tool will provide the user with a list of search results, including a definition for every term, and then the user can just select which sounds the best for him. And the annotations we got from the users are currently only used for a text search for the exact ontology terms and for synonyms. So, like I mentioned before, the current search in hPSCreg is text-based, and we are finding more or less only exact results. And the most problematic is that it's based on user-entered keywords. So if you have two users describing the same thing, they tend to use different words, and therefore it's quite difficult to find the cell line you want to have. And even more people currently are used to find more intelligent results than just exact results. So if somebody is searching for a cell line from a patient with cancer, he would also like to find cell lines from a patient with retinoblastoma, which actually is a kind of cancer. And since ontologies can effectively be used to assign meanings to words so that the computer knows that the retinoblastoma is a form of cancer, I had the idea to use kind of an hPSCreg ontology to improve our search results. And as it doesn't make much sense to create a new ontology from the scratch, I got in contact with the team of the cell line ontology, and together with Oliver from the cell line ontology, we decided to import the data from hPSCreg into the cell line ontology. So I guess most of you will be familiar with the cell line ontology, but I just want to give a very brief introduction. The cell line ontology is an open biological and biomedical ontologies library ontology about cell lines, And the goal of the CLO is to standardize the representation of cell line cells around the world. Like hPSCreg, the cell line ontology is a community-based project with a lot of partners. And the cell line ontology has first been released in 2010 with data from ATCC, the Coriell Institute, and HyperCLDB. And in 2014 there was a large update with information with cell lines from the Riken Institute, and another large update was performed in 2017 with data from the Links data portal and the Chinese Academy of Medical Science. And since end of 2018 we are working on importing the cell lines from hPSCreg into the cell line ontology. So up to today, the cell ontology contains more than 40,000 terms describing more than 38,000 cell lines, and terms are imported from more than 20 ontologies. So for example, we have 134 cell types, more than 880 diseases, 400 anatomical entities, and 317 taxonomy terms which are imported from other ontologies. Before we could start to import the data from hPSCreg into the CLO, we first had to think about which information from hPSCreg could be really represented in an ontology. And after some discussions we came up with the list presented here. So we have some basic information on stem cells, which includes the standard hPSCreg name, the cell type of the source cell, the origin of the source cell, and the parent term. So whether it is an embryonic stem cell or an induced pluripotent stem cell. We also have information on alternative terms and database references, and we have subclones, which are genetically modified cell lines. And for these kind of cell lines we have of course a name and the name of the cell line it has been derived from, and again the information on the parent term. And we further want to describe the genetic modifications in more details, including the type of the modification, so whether it is a gene knockout, a gene knock-in, an isogenic modification or just transgene modification, and of course the affected gene. And in some cases such genetic modification acts, and so the cell line with this genetic modification then acts as a disease model. And as the cell lines have been derived from a donor, we also want to include the information on the donor. So for each donor we provide a so-called biosamples donor ID. The biosamples database is hosted from the EBI, and it's a unique ID for cell lines and donors, which provides a connection across different databases. And furthermore we have the information for donors about the genetic sex, and we have other synonyms used for the donor and more database cross-references. Furthermore we have information on the disease of a donor, and we have information on relations among different donors. So in some cases we have family control lines, meaning that we have two relatives, and for example one of the relatives is affected by a disease and another is healthy. To import the data from hPSCreg to the cell line ontology, we wanted to use the ontology tool from the He group, from Oliver He, because there is really a large number of cell lines in hPSCreg and we don't want to do this manually. And the development from ontology is based on the ontology design patterns. It has been inspired by the OB QuickTerm templates and implements OB QTT procedure, and it has a web-based interface. And to use ontology, there are 3 parts needed for the execution. So first you have to define the target ontologies to avoid ID duplications. you have to have the input data file, so in Excel or in tab delimited text form, and you have to have defined the settings. It could be done with a script, and the settings can be stored and reused. And after you run the tool, you get an OVL file, output file, and you can also get an Excel sheet or a tab delimited text file that could be then imported again in a database to have the ontology IDs in a database. And this tool is freely available online at ontology.hgroup.org. And yeah, here's what it's look like. So you define, if you already have some setting files, you can upload them, and then you have to specify the target OVL file, and then you have to specify the settings, so what new axioms will be used to generate new class or to update classes, for example, and you have to define the annotations and equivalent classes, superclasses, and other terms to define the annotations. And then you click on Get OVL, or you can save the settings first, and then you get this OVL file. So to import the cell lines from hPSCreg into the cell ontology, we had to run different steps. So first of all, we had to define the design patterns for the export. Then we of course had to export the data from hPSCreg into this specific design pattern. Then we are using the ontology tool to create an OVL file, and then we also need to import the ontology IDs created by ontology into hPSCreg again. And as we have a lot of information in hPSCreg will depend on each other, we had to run these steps several times. So first we begin with standard cell lines, and then we have the IDs for the normal cell lines, and then we could also export, for example, genetic modified cell lines based on these cell lines. So how do these design patterns look like? So first we manually created the design pattern to just define what are human embryonic stem cell line cells and human induced pluripotent stem cell lines. So both are derived, of course, from the Homo sapiens, and an embryonic stem cell line cell is an embryonic stem cell line, and an induced pluripotent stem cell line is an induced pluripotent stem cell. Both are pluripotent, and both are stem cell line cells which are capable of stem cell division and cell differentiation. And after we had this basic up-level framework, we thought of how to represent a specific cell line. So we started with the cell line, the BHI005-A cell, and very important is we decided to create donors not as classes but as instances of the type human tissue donor. So here we have a human tissue donor, and we use the biosamples ID, which always looks like this, so with some year and then a long number. And in this case we have a male tissue donor, and then we know that this cell has been derived from a fibroblast of the dermis, from the papillae of penis, and this is a human-induced pluripotent stem cell line cell which has been derived from a Homo sapiens. Yeah, so this was a very easy example. Like I mentioned before, we also have familiar control lines. So here we have an example with 3 tissue donors. One tissue donor is suffering from unipolar depression. It is a male tissue donor. And this one is an uncle of another tissue donor who also suffers from unipolar depression. It's also a male donor. And he has a brother who he's not suffering from unipolar depression, he's healthy. And from all of these 3 tissue donors, there have been derived 3 cell lines. So here we have the ED014 ABC, ED15 ABC and ED16 ABC. And I mentioned before that we also have subclones, so genetic modified cell lines, and we also had to think about how to represent them. And here again we have the cell line BHI005-A, which derived from this donor. And then we have two cell lines which have been genetically modified. So here we have the BHI005-A-4 line, which has been derived from the parent line with the knockout of the gene SORT1. And the knockout of this gene could also then, the cell line could then be used as a disease model for myocardial infarction. And yes, we could use this for several modifications of a cell line. After we have designed the basic design patterns, we could start with the actual import process. And as we are using the tool ontology for the import, we had to take several steps for importing to consider the technical dependencies. So first of all, we were importing the basic stem cells, which are not genetically modified. And afterwards we imported the genetic modified subclones. And then we define the modification details. So we start with knock-in genes, knock-out genes, isogenic modifications and transgenetic modifications. And afterwards we imported the disease models. And then we had several ontology files, and then we merged all these OVL files to one file, and afterwards we could import the donor information. And we had to do the import of the donor separately because the tool ontology only supports classes, but donors are designed as classes. Therefore we can't use ontology for importing. And thankfully Edison Ong from the He group first writing some scripts to automatically import the donor information as instances into the OVL file. Further problems that arise due to the import of the cell lines were that the ontology terms in hPSCreg are annotated by users, and therefore they use several ontology terms for the same thing. And the CLO usually imports only specific ontologies. So for example, they used the disease ontology for diseases, and therefore a mapping was needed. And we tried to do this semi-automatically, so we had to check everything manually. And in the end, we were quite successful, and we could import 2,372 hPSCreg lines from hPSCreg into the CLO, and the OVL file is all public available. 1,500 of these cell lines have information on the source cell type with them, and for around 1,000 we have information on the anatomical origin of the source cell. For 43 subclones, information on the type of genetic modification is available. This is quite few because currently this information is mostly available only as a free text, which modification has been done, and therefore we can't map it yet to an ontology. Furthermore, we exported more than 1,000 donors, 811 of them are associated with a disease. Overall, we had more than 80 different diseases, and of these 81 diseases, 58 could be mapped to a disease ontology term and imported into the cell line ontology. And then we had a nice overview of the cell lines in hPSCreg and the cell line ontology with all the annotated properties. And so our next step would then be to provide a SPARQL endpoint in hPSCreg to allow semantic queries. And we also want to include the CLO in the search interface of hPSCreg to allow semantic queries not only using Sparkle but really including it in our Elasticsearch routine to allow free text search for the ontology terms. And another important step would be to implement some kind of an update mechanism to update the CLO with hPSCreg data on a regular basis, as we are getting new cell lines continuously. So currently it's around 20 to 40 new cell lines every month that are validated in hPSCreg. Yes, and with that I want to thank you for your attention, and I want to thank the people who work together with me. So from the CLO team we have Professor Oliver He and Edison Ong, and from the hPSCreg team we had our group leader Andreas Kurz, and I have my colleagues Johannes Dibender, Nancy Ma and Niels Boudier.